In a world increasingly dominated by wireless technology, concerns about the potential health effects of radiofrequency radiation from cell phones have sparked intense debates. Central to these discussions is the question of whether findings from animal studies are relevant to human health. Recent groundbreaking research provides a compelling counterpoint to skeptics, highlighting morphological similarities between tumors found in rats exposed to radiofrequency radiation and low-grade human gliomas. This discovery underscores the importance of considering animal study results when assessing the potential health risks of radiofrequency radiation exposure. The National Toxicology Program's landmark study. The National Toxicology Program, a division of the National Institute of Environmental Health Sciences, conducted one of the most comprehensive studies to date on the potential health effects of exposure to cell phone radiation. Released in 2018, the study exposed rats to radiofrequency radiation, similar to that emitted by cell phones. The results were alarming. Male rats exposed to radiofrequency radiation had a higher incidence of tumors, with 1 in 12 rats developing malignant or pre-cancerous lesions. This translates to a probability of approximately 8.3%. Key findings: tumor development. The tumors were primarily located in the brain and heart, mirroring the organs affected in certain human cancers. Nonlinear dose-response relationship. The study found that lower doses of radiofrequency radiation resulted in a greater incidence of tumors, suggesting a nonlinear response. Exposure levels: rats were exposed to radiofrequency radiation levels below current regulatory limits for human exposure, prompting questions about the adequacy of these safety standards. Implications. The National Toxicology Program study's findings challenge previous assumptions about radiofrequency radiation safety. The observation of tumor development in 1 out of every 12 rats is significant, especially considering that no other study has shown such a high incidence of cancer in rats from exposure to a substance later deemed safe for humans. The Ramazzini Institute's corroborative research. Adding weight to the NTP's findings, the Ramazzini Institute, or RI, in Italy, conducted a parallel study examining the effects of cell phone radiation on rats. The RI study exposed rats to RFR at levels much lower than those used in the NTP study, yet the results were remarkably similar. Key findings. Rats developed the same types of cancer as those in the NTP study, including heart and brain tumors. Rats were exposed to RFR starting in the womb and continuing throughout their lives, totaling 18 hours a day. Even at levels below the legal limit for human exposure, which is 1.6 watts per kilogram of body weight, significant tumor development was observed. Implications. The RI study's findings are particularly concerning because they suggest that even low-level chronic exposure to RFR can lead to cancer development. The replication of tumor types between the NTP and RI studies strengthens the argument for a potential link between cell phone radiation and cancer. One of the most compelling aspects of these studies is the morphological similarity between the tumors developed in rats and low-grade human gliomas. Gliomas are a type of tumor that occurs in the brain and spinal cord, arising from glial cells that support nerve cells. The translational relevance of these similarities suggests that the biological effects observed in rats could be reflective of potential effects in humans. While species differences are a factor in scientific research, such parallels provide a strong argument for the relevance of these findings to human health. This evidence counters the argument that animal studies are irrelevant to human health concerns, particularly regarding cell phone radiation. The human relevance of these findings is further underscored by real-world cases like that of Jimmy Gonzalez, a Florida attorney who developed tumors in both his brain and heart, the same organs affected in the NTP and RI rat studies. Before his passing, Gonzalez became an advocate for raising awareness about the potential health risks associated with cell phone radiation. His case aligns with the types of tumors observed in the animal studies, suggesting a possible link between prolonged cell phone use and cancer development in humans. Gonzalez's story has been cited in discussions about the need for precautionary measures and further research into RFR exposure. A notable aspect of the NTP study is the non-linear dose-response relationship observed, where lower doses of RFR resulted in a greater incidence of tumors. This suggests that biological effects of RFR may occur without significant heating of tissues, pointing to bioelectric mechanisms rather than thermal ones. Current exposure limits are based on thermal effects, and a non-linear response challenges these standards, indicating that even low levels of exposure may not be safe. This finding emphasizes the need to explore the underlying mechanisms of RFR's biological effects at various exposure levels. Real-world exposure considerations. The NTP study utilized a 10-minute on-off cycle for RFR exposure, totaling 12 hours a day. However, real-world cell phone use often exceeds these intervals, with longer durations and constant body contact. Factors to consider: the advent of 5G technology and the proliferation of cell towers and satellite-based communication systems have increased potential exposure. Continuous exposure from early life stages, similar to the lab rats, is becoming more common in humans. Many people may be exposed to RFR levels that exceed those used in the studies, raising concerns about current safety guidelines. Gender-specific effects. Both the NTP and RI studies observed a higher incidence of tumors in male rats compared to female rats. Relevance to humans. Human studies have also found a higher incidence of certain brain tumors in males. Hormonal differences, genetic susceptibility, and lifestyle factors may contribute to gender-specific effects. Understanding these differences is crucial for developing gender-specific guidelines and protective measures. Implications for public health and safety standards. The findings from these studies have significant implications for public health policies and safety standards related to RFR exposure. Calls to action. The evidence suggests that current regulatory limits may not be sufficiently protective, necessitating a reassessment of safety standards. Encouraging the use of hands-free devices, limiting call duration, and keeping phones away from the body can reduce exposure. Increased awareness about the potential risks and safe usage practices is essential. Debate and skepticism. Addressing counter-arguments. Despite the compelling evidence, skepticism remains regarding the applicability of animal studies to human health. Common counter-arguments, species differences. Critics argue that physiological differences between rats and humans limit the relevance of the findings. Exposure levels. Some suggest that the exposure levels in studies do not accurately reflect typical human usage. Responses, morphological similarities. The observed similarities in tumor types and locations strengthen the argument for relevance. Realistic exposure scenarios. The studies used exposure levels comparable to or below those experienced by heavy cell phone users. Historical precedents. No study showing such a high incidence of cancer in rats has later been proven irrelevant to humans. The need for continued research. The studies underscore the importance of ongoing research into the potential health effects of RFR exposure. Future directions, long-term epidemiological studies. Monitoring health outcomes in populations with varying levels of exposure can provide valuable data. Mechanistic studies. Investigating the biological mechanisms of RFR-induced carcinogenesis can enhance understanding. Technological innovations. Developing and promoting devices with lower emission levels can mitigate risks. Global perspectives and regulatory responses. Different countries have varied approaches to regulating RFR exposure. Examples: precautionary policies. Some nations have adopted precautionary measures, especially concerning children's exposure. Regulatory updates. Agencies may need to update guidelines based on emerging evidence. International collaboration. Global efforts can harmonize standards and promote research. Personal actions to reduce exposure. Individuals can take proactive steps to minimize their exposure to cell phone radiation. Recommendations include using hands-free devices, Utilizing speakerphone or earphones keeps the device away from the head. Limiting call duration. Shortening calls reduces cumulative exposure. Avoiding body contact. Refrain from carrying phones in pockets or close to the body. Texting instead of calling. Text messaging emits less radiation than voice calls. Industry responsibility and innovation. The telecommunications industry plays a crucial role in addressing these concerns. Opportunities include design improvements, developing phones with lower radiation emissions, transparency, providing clear information about emission levels and safe usage, research investment funding independent studies to explore health effects. Balancing technological advancement and health. As society becomes more dependent on wireless technology, finding a balance between innovation and health safety is imperative. Considerations include informed choices. Users should be empowered with knowledge to make decisions about device usage. Regulatory oversight. Governments must ensure that technological progress does not come at the expense of public health. Ethical responsibility. Companies should prioritize user safety alongside profitability. Conclusion. The convergence of findings from the NTP and RI studies, along with morphological similarities to human gliomas, presents a compelling case for reevaluating the potential health risks associated with cell phone radiation. While acknowledging the limitations inherent in translating animal studies to humans, the evidence suggests that precautionary measures are warranted. The observed incidence of tumors in 1 out of every 12 rats exposed to RFR is unprecedented in scientific research, especially considering the similarity to human cancers and the exposure levels involved. As wireless technology continues to permeate daily life, it is crucial to prioritize research, update safety standards, and promote practices that minimize potential risks.